Okay, hey everybody, welcome back. My name is Andy Horgan, and I am another member of the committee. First of all, if you're just now tuning in, thank you so much for joining us today. I think we have some great stuff coming our way in the next hour. So with that, I'm going to introduce our next speaker. Dr. Elizabeth Maga is an associate professor of applied molecular genetics in the Department of Animal Science at the University of California, Davis. She has a bachelor's of science in chemical engineering from Colorado State University and a PhD in food science and technology from UC Davis. Her research focuses on applying biotechnology to animal agriculture as a tool to improve animal and human health. Her current work is directed at translating the use of lysozyme-rich milk from genetically engineered goats to fight intestinal diseases and the generation of gene-edited pigs for agricultural and biomedical purposes. Dr. Maga has taught courses in molecular biology, introductory animal science, and advances in animal biotechnology, as well as lactation and integrative animal biology at UC Davis. She has received multiple federally funded research grants to study the impact of transgene presence and expression on the biology of the transgenic animal, and a Grand Challenges Exploration Award from the Bill and Melinda Gates Foundation to study the effects of milk on malnutrition using a pig model. So with that, I'm going to hand the mic over to Dr. Maga, and let's all welcome her, and it's all yours. Take it away.
Thank you very much, Andy. And I'd like to thank the organizers for inviting me to present work today that I have been doing for a very long time now. Hang on. And just sort of a spoiler alert. I'm not going to be talking about gene editing really. So I'm going to be talking about an application of the original type of gene editing, which is straight up transgenesis. So we'll be talking, as Andy said, about studying the milk from transgenic goats to fight diarrheal illnesses. So the whole root of my work that I'm going to present today revolves around the problem of malnutrition and diarrhea in young children. So while there has been a lot of progress made over the years, there is still over 5 million children under the age of 5 who died in 2018. This is the most recent data year we have. And of these deaths, children beyond the age of breastfeeding, diarrhea remains a leading cause of death. So almost 8% of these deaths of under 5s was related to diarrhea, and over almost half of these childhood deaths are related to malnutrition. So we know malnutrition early in life, in the first 1,000 days, can lead to deficits in cognition and metabolic dysfunction later in life. And malnutrition damages basically the state of the intestine. And so when you have a malformed intestine, then you're more susceptible to pathogens that can cause diarrheal illnesses. And likewise, if you get a pathogen that causes a diarrheal illness, that can further damage the intestine and make it unable to absorb the nutrients that the person is eating. And so that links these two diseases in this really sort of vicious cycle. So for children of the age of who are breastfeeding, breastfeeding is recommended as one of the tools to combat diarrhea. But for children beyond the age of breastfeeding, animal milk, like from goats and cows, it lacks these protective proteins that is present in human milk. So that makes the milk of dairy animals not as suited as human milk is to help protect infants from diarrheal causing infections. So when we embarked on this work many years ago, we hypothesized if we could engineer the milk of dairy animals to contain these protective factors of human milk, we could maybe have a tool to help prevent diarrhea in children beyond the age of breastfeeding. So just a little bit about these protective proteins in milk. There's a set of them that play a role in protecting the infant from infection. And also they play a role in developing the set of microbiota that inhabit the intestinal tract. So there have been a lot of studies that have shown the types of bacteria in the intestinal tract of infants that get breastfed is distinct from those who get formula fed. And that can also, that difference in microbiota can also be related to the ability of the infant to fight off infection. So they have less incidence of diarrheal and respiratory infections. So the microbiota is playing a really big role in the infant. And so in human milk, there are these bioactive, what we call protective factors, such as lactoferrin, oligosaccharides, IgA, and lysozyme. So lysozyme is miraminidase. It's an enzyme. It's found in the tears and saliva and milk of all mammals. And it's part of the innate defense system. So lysozyme is an enzyme, and what it does is it specifically cleaves a link in the peptidoglycan of the cell walls of bacteria. So that causes them to leak, lyse, and not be able to grow as well. And just when we talk about milk, I've noted here, we see a big difference in the levels of lysozyme in the milk of human milk and of that of our dairy animals, such as the goat and the cow. So there's almost 3,000 times more lysozyme in human milk than in cow milk, and almost more than 1,500 times more than goat milk. And in human milk, this level of lysozyme starts low at birth and then increases and stays at this high level throughout the duration of lactation. And in goats and cows, lysozyme levels are actually very high when the animal gives birth. And then at the end of lactation, when the animal is drying off, and also when the animal has an infection, lysozyme levels go up. And that's because lysozyme is part of the natural defense system. And at birth and involution, when the animal's drying off, those are the times when the animal is most susceptible to infection. So because of these physiological differences between species, it's not possible to select goats or cows that naturally make high levels of lysozyme in their milk throughout lactation. So to sort of overcome this physiological difference, we used genetic engineering to make a goat that expresses human lysozyme in their milk. And our hypothesis is if this lysozyme is present in the udder of the animal, we can envision several benefits to the animal and to consumers of the milk. So if the lysozyme is there, it should be able to modulate the types of bacteria present in the gut and the udder. So we could envision an animal that has more resistance to mastitis. So mastitis is an infection of the udder, which is quite common in dairy animals, and it's a huge welfare problem. So we maybe, if lysozyme is present in the udder, in the milk, we could decrease the incidence and severity of mastitis. The lysozyme could also influence the safety and longevity of the milk. It could help prevent maybe perhaps the spoilage organisms from growing in milk. And if an animal or a human would ever eat this milk, maybe you could act like the lysozyme does in human milk to modulate bacteria and help fight or prevent infections. And furthermore, having lysozyme in the milk of an animal, so in a food product, should be safe because, as I mentioned before, lysozyme is present in many secretions in humans, such as tears and saliva. So basically, every time you're swallowing, you're eating lysozyme. So if you were to eat human lysozyme, it's being made in the milk of a dairy animal, it should be safe. There shouldn't be an allergic reaction. So taking this sort of background into mind, we started many years ago now on this journey. I'm going to call it a journey or a story of how we started with using mouse models to see if we could express lysozyme in the mammary gland. What would that do to the functionality of the milk? So we were the first group to show that we could use a transgenic approach to alter the functionality of milk. So our mice, we got to milk mice, it was very exciting. They made lysozyme in the milk, it was active, and it could, in vitro tests, it could slow the growth of several different types of bacteria that are responsible for milk spoilage. So because we had our proof of principle that we could make active lysozyme in the mammary gland, we moved on and we generated transgenic goats. So when we started this work, we were using the goat as a model for a cow. So basically a goat for us was a cheap cow. Goats also have a shorter gestation interval, 5 months versus 9 months, and they have multiple offspring. So this means we could generate more data in an efficient manner. So since these goats have been born, we have been doing two arms of research with them. One line of research is we've been looking at risk assessment. So because these goats have an extra piece of DNA in them, carrying our lysozyme transgene, are they in any way different from any other goat? So I'll describe to you a little later how we're sort of doing some risk assessment type studies. And then the translational piece of the work is, can we take the milk from these goats and use it to help treat and prevent diarrheal illnesses, right? So to do that, since our target would be young children beyond the age of breastfeeding, we're using the pig as an animal model to see what happens. So the overarching hypothesis over the use of the milk would be, we feed the milk to an animal model, it should modulate the types of bacteria in the gut, and that should make the animal more healthy and resistant to diarrheal causing infections. So I'll describe to you some work today where we have worked with healthy pigs and pigs that we have induced diarrhea in, and then also our work with the malnourished animals. So all our models that we're doing, basically we're trying to answer the question, how does lysozyme in the milk affect the microbiota of the animal, and how does that influence the host response? And we're taking a systems biology approach. So basically we look at the effects at the whole animal level. So the physical characteristics of the animal, the phenotype, if you may, and then we take a deep dive, and we're really interested in what's going on in the intestine of the animals. And with that, we're interested in the types of bacteria that are inhabiting the intestinal tract. The intestinal epithelial cells themselves, are they influenced by the milk? And then is the milk actually causing any changes in gene expression, and how are all these things related to each other? All right, so some background on our line of transgenic goats. So our lysozyme goats were made by pronuclear microinjection. So we take a 1-cell zygote. So this is a fertilized egg, and we have to do micromanipulation. Not as easy as working in the butterflies as we heard in the other talk, but we inject our transgene DNA, which in this case was a bovine alpha S1 casein transgene. So we have the cDNA for human lysozyme hooked up to regulatory elements of a casein gene. So caseins are proteins that are only made in the mammary gland and only made during lactation. So the goal here was to have the expression of the lysozyme restricted to the udder of the animal and only when the animal is lactating. So from our microinjection experiments, we resulted in one founder animal who is Artemis, who is shown here. And so all the work I'm going to talk to you now about the goats comes from Artemis and descendants of her. So we're in the probably 9th generation of animals that have come from our founder animal, Artemis. So in terms of lysozyme expression, so the lysozyme is expressed in milk at 270 micrograms per mil. So this is about 68% the level found in human milk, and it's over 1,000 times more than is normally found in goat milk. These are Western blots over here. We can show that we have consistent expression of the lysozyme between individuals and across different parities, so successive lactations. And if we take milk over time, or pooled milk from all our transgenic animals, and we look over time, we can see we have consistent levels of expression of our lysozymes compared to our control non-transgenic goats. The lysozyme in the milk is active, so we can do, these are spot-on lawn assays. So we put a lawn of micrococcus on an agar plate and we drop milk on it. And where you see the black, here's the clearing, that's where the lysozyme has eaten away the bacteria. And we can show, we have done a little work to show that the lysozyme made in the mammary gland is a little more active than purified lysozyme you can purchase. So hen egg white lysozyme is commonly used in the food industry and the cosmetic industry as a preservative. You can see if we spike the hen lysozyme into the milk, we don't get as much clearing as the milk made in the udder itself. So we think that has to do with maybe the other components of the milk that are there, or the types of post-translational modifications that the mammary gland can do. The overall gross composition of the milk between transgenic and wild type animals is not different by doing standard protein and fat testing monthly with our animals. We also took a deeper dive and did some two-dimensional gels and mass spectrometry testing. And we saw there was no gross disruption of the protein composition. So all the proteins we found were milk proteins, and they weren't up or down regulated in any different levels between our transgenic and our control animals. So the only difference in the composition between the milks is the presence of the lysozyme. So some of the risk assessment work that we've been doing, the first is just basic, are the animals healthy, right? So we did a study where we monitored the lactation of the animals over time, the behavior, their reproduction, and the growth. So we looked at the growth of animals that were raised on lysozyme milk or control milk, the growth of the adult animals, basically all these basic parameters we looked at. We found no significant differences between the transgenic animals and the wild type controls. We've also been looking at the safety of the products. So our transgenic animals carrying the gene's only expressed in the milk. So if we look at the milk, we're looking at the microbial profile. So because we have this antimicrobial in the milk, did we anyway really skew what type of bacteria are there in a good or bad way? So we can see, just looking at the main bacteria that we found, or the main phyla that we found in our milk samples, you can see they change over time between early and mid and late lactation. So early lactation for us is the spring, this is usually the summer, and late lactation is now in the fall. And these are for our control animals. For our transgenic animals, they followed the same patterns, basically. So we didn't alter the microbial profile of milk to a great extent. And then we also were trying to look at, so this is our target organ, right, is the mammary gland, but we also want to just try to do some assessment of looking at if there's any sort of off-target effects of having animals express lysozyme in the udder. So we took serum from lactating animals and did a metabolomics study. So we tried to identify and quantify the metabolites present in lactating transgenic and lactating non-transgenic animals. Basically, on the PCA here, you see there's no clear pattern between transgenic and control animals. Between the hundreds of metabolites we identified, one was significantly different between transgenic and controls, and it was an unknown. So from this work, we can conclude that the presence and expression of the transgene is not detrimental to the overall function of the animal, and it's not altering anything in an adverse way. So a little bit more on the antimicrobial activity of the milk. So we can take the milk from the transgenic animals and incubate it with isolates that cause mastitis. So E. coli and Staph aureus are main culprits of mastitis. And also ones that cause the spoilage of milk. So a Pseudomonas fragile. So if you ever leave your milk in the refrigerator too long and it starts smelling gross, this is the guy responsible for that. And in all cases, the lysozyme in the milk could slow the growth of these organisms. If we look at sort of a practical application of this, so if we took milk from a transgenic animal and a control animal and set it out at room temperature for a couple days, and then we would plate it, plate the milk on an agar plate and see what would grow. You can see the lysozyme milk. This is 2 nights out of the refrigerator. We have no growth, and you can see in the non-transgenic control milk, we have a lot of bacteria growing. So our milk has a longer shelf life. And people always ask, well, can you make cheese with the milk? Because lysozyme, you need bacteria to make cheese. So I have a student, Erica, here, who showed that indeed you can make cheese with the lysozyme milk, and the bacteria, the lactic acid bacteria you use to make the milk, they do start degrading the lysozyme milk. They do start going away a little earlier, but that could be a good thing, because when that happens, that releases all the enzymes that are going to help develop the flavor and texture of the cheese. We also showed that you could make cheese quicker. So the time it took the curd to form was quicker, and the packing of the cheese, or the strength of the curd that formed, was a little stronger with the lysozyme. And that's because lysozyme also has another property. It has a net positive charge, which allows it to do all these different types of physical chemical interactions with the milk proteins. So the lysozyme is there. It's active in the milk. So what happens then if we would feed the milk to an animal? And as I mentioned before, we used a pig as a model animal, and that's for a couple reasons. One, the main reason is sort of depicted in this little cartoon here. The digestive system of the pig is much more similar to humans than our commonly used rat and mouse models. So when we talk about doing translational research, we want to make sure we use a correct animal. So the pig is a much more human relevant animal model, especially when you're talking about diseases of the intestine. So in our proof of principle type of experiments, so remember our overarching hypothesis is if we feed our animal model the lysozyme milk, we should modulate or change the types of bacteria in the gut, and that should make them more resistant to infections. So to start with, we started with, and again, remember our model is for children beyond the age of breastfeeding. So we start with 6-week-old pigs. So pigs get weaned at 3 weeks of age. And we fed them pasteurized milk from transgenic or control animals twice daily for a period of 2 weeks. And we investigated, we looked at types of bacteria in their gut, we looked at the structure of the intestine, and we did standard blood work to assess what eating the milk would do to the pigs. So we started simply by looking at the total coliforms and E. coli that were present in the upper and lower small intestine of the animals. And we saw reductions in the number of coliforms and E. coli in the upper lower intestine. So this was our first evidence that consuming the milk could change the types of bacteria that were present along the length of the gastrointestinal tract. We looked more in depth then, instead of just culturing, we started using molecular techniques to look at what type of bacteria were there. And so what we saw was that all the animals that were fed the lysozyme milk had a similar microbiota composition. This is in their feces. And they were totally distinct from the animals that got fed regular goat milk. And so what we learned was that we basically, the lysozyme milk enriched for bacteria that are considered biomarkers of gut health. So these are like bifidobacteria. et cetera. And it also reduced bacteria associated with diseases. So again, so our firmicutes, this is where the Clostridia and stuff live, the phylum, and our back went down and our Bacteroides went up. So we had this shift in the bacterial composition in our gut. So our first sort of main hypothesis we were testing was, yes, the lysozyme milk can modulate intestinal microbiota populations. We were also interested to see what that would mean in terms of the structure of the intestine. So here we have, so in the intestinal tract is lined with these villi, these projections that reach out into the lumen, and they're there designed to absorb all the nutrients, take up all the nutrients as they get broken down as they move through the intestinal tract. So we can measure the height of these villi, and the depth of the crypts next to adjacent villi, how thick the lamina propria is, or the part that's attaching it to the muscle layer of the intestine. And we can also look at lymphocytes that are living in the villi, as well as these mucin-producing goblet cells. So when we looked at the impact on the intestine, the lysozyme-fed animals tended to have longer villi. They had a thinner lamina propria, so this correlates to more absorptive surface area for the animal. We had fewer lymphocytes within those villi, and we tended to have more goblet cells. So this would indicate these goblet cells produce mucin, and that's one of the protective barrier layers in the gut. So this to us says it's the gut is in a more quiescent state. We also looked at expression in the tissue of different anti and pro-inflammatory cytokines. So we had an increase in TGF, which is a anti-inflammatory cytokine. Our pro-inflammatory ones were not changed between transgenic and control. And in terms of the blood work we did, there were no significant differences in any of the blood cells. And I just want to point out, I have listed here eosinophils. That's one of the main drivers. If an animal has an allergic reaction, those would be askew. So basically, the milk did not cause any sort of inflammation in the animal or an allergic response. So that was, we had our proof of principle model in a healthy animal. So the milk could shift bacteria and the animal tolerated it, and we had an impact on the state of the intestine. So for our next studies, we wanted to go on and look in a disease model. So we used an enterotoxigenic E. coli. So ETEC are one of the main causes of diarrhea in young pigs. When you wean a pig, they undergo a lot of stress and dietary changes. And so enterotoxigenic E. coli are one of the main causes of diarrhea. It's also a main cause of diarrhea in humans as well. So we developed two models, a treatment model and a prevention model. So again, we start with our 6-week-old pigs, and in our treatment model, we would dose them with our ETEC to induce diarrhea. And then we fed them milk. So this is a model where if an individual is sick, if they drink the milk, will that help them recover from the illness? And our other model was a prevention. So in this model, then we fed the animals milk for a period of 2 weeks, and then we challenged them with E. coli to see if the milk was so protective or not. And again, we have our whole list of parameters that we investigated. So in our treatment model, we found that the lysozyme-fed pigs were able to recover quicker from the diarrhea than our control-fed pigs. So if we look at our graph over here, so all our pigs were challenged with E. coli at the starting point. And then about 30 hours after challenge is when we started feeding them milk. So we're looking at the fecal consistency. So we can see as time goes on, the fecal consistency goes down, meaning the animals are having active diarrhea at this point. So we start feeding them the milk, and we can see the animals that got fed the lysozyme milk recovered quicker. Their fecal consistency got better quicker than the animals that received regular goat milk. Our blood work also indicated that the animals in the lysozyme group were less dehydrated. They cleared their infection faster, so the leukocytes went back to normal levels, while the ones in fed control milk were still recovering. And we can see in our intestinal morphology over here, the lysozyme-fed animals had really nice villi, and their lamina propria wasn't very inflamed. And over here, this is a picture from a control-fed animal, and you can see that it's very different, and so that this intestine is still recovering from the infection. So this was very exciting for us to actually show the milk could help in one of these situations. So then we went on to look at our prevention model. So if we fed the milk to an animal first, would they be protected from diarrhea? So basically we did this study in both healthy and malnourished pigs. So again, we would start at 6-week-old pigs, feed them milk twice a day for 2 weeks, and then challenge them, and then a few days later do the necropsy. In our malnourished model, it's the same sort of layout, except we started with weaned pigs, and we fed them a restricted diet for 3 weeks before we started the milk treatment. So I'm going to go and outline our models really quickly. So for our malnourished study, we fed our weaned pigs a protein and calorie restricted diet for a period of 3 weeks. And we had our full fed group that got standard ad libitum access to their standard feed. And so after 3 weeks, we necropsied several of our animals because we wanted to see if we were having changes that were indicative or that could recapitulate the human situation of malnutrition. So after 3 weeks, our animals in our malnourished group weighed about half as much as the full-fed animals. And you can see here we had numerous differences in blood chemistry and blood cells. They were all indicative that we have induced a state of malnourishment in these animals. We also looked at the intestinal structure of these animals, and the intestine was much smaller in surface area, so shorter villi, thinner villi, and we also did some in ex vivo work to see how the flow between cells was. So if the intestine is damaged, the flow both between and within a cell is sort of messed up. And so we can measure this with an Ussing chamber. And so we could see that the junctions between our intestinal epithelial cells were damaged in our malnourished model because we had increases. We tended to have increases, and we had a significant increase in the movement of molecules between cells. So taken as a whole, this showed that our approach to feeding a reduced calorie and protein diet sort of recapitulates many of the aspects of malnutrition that are seen in human children. So and one more thing that we showed, that the restricted diet also shifted the types of bacteria in the gut. So here you can see we have distinct compositions of bacteria between full-fed and malnourished animals. So there are different families that, for instance, we were enriched in enterobacteria. So this is where E. coli lives. And also it was interesting to note that this family called Christianelliceae, which is associated with lean body mass, was also increased in our malnourished animals. Okay. So we were convinced we had a working model now. So we went on and tried to see what would happen if we fed the animals goat milk or milk from our lysozyme transgenic animals. So we fed the milk for 2 weeks. The overall weight gain was not significantly different between just the no milk group and our 2 goat milk groups. However, the rate of gain was greater for both the goat milk and the lysozyme group. And then when we measured the permeability, the ex vivo permeability in our intestinal sections, we can see that the goat milk helped reduce it, and then the lysozyme milk helped reduce it even more. So sort of the trends we saw throughout all of our parameters that we're studying, goat milk helped improve the state of malnutrition, and the lysozyme containing goat milk helped it a little more. So again, here's some pictures of what the intestine looked like. So this is our malnourished animal with no milk. The goat milk had improvements in villi height, and the lysozyme milk again had a little bit greater improvement in these parameters. The milk also, like as we showed in our healthy model, it also shifted the types of bacteria that were present in the gut. So in this PCA plot, our malnourished animals are clustered here, and you can see the goat and the lysozyme milk change the bacteria, but in a different way. So it's having differential effects on the types of bacteria that are present in the animals. So as the final stage of this study, then we had a subgroup of animals still left that we challenged with E. coli to see if feeding the milk could help prevent the infection. So what we saw during infection was the milk actually played a very important role in helping the animals maintain their weight gain during the infection, during the challenge. And we can see that the lysozyme milk helped a little more. So the animals weighed or gained more weight than animals in the goat milk group did. And the lysozyme milk was significantly different from those that got no milk during the challenge period. So this is an important aspect of just pigs in production. If they keep gaining weight, even while they're sick, that's a good thing. So in terms of protecting against infection, the lysozyme milk was effective at doing that in both healthy animals. So these are animals that weren't malnourished first. We can see, again, these are looking at the fecal consistency. So the animals had their milk for 2 weeks, they were challenged, and you can see animals that got fed regular goat milk had more severe diarrhea than those that had the lysozyme milk. And with our malnourished study, we had a fewer numbers of animals, but we did see trends. So you can see our malnourished animals, their fecal consistency continued to decline 2 days after challenge, and the goat milk didn't really help too much, but the lysozyme milk, the fecal scores never got as bad as the other 2 conditions. And again, we saw differences in the intestinal structure during the challenge period. So taking all these physical characteristics of our animal now, we're moving on to try to see how is the lysozyme milk causing all these changes to happen. So is it just affecting the bacteria, and then the bacteria are doing all these other things with the metabolites and everything they produce? Can the lysozyme milk act directly on the intestinal epithelial and change gene expression? Or is that, or are the bacteria and the genes, are they all talking? What's the crosstalk going on? So what basically is driving all these changes that we're seeing in our animals? So we've been looking, we're taking an omics approach to this. So we have data on the transcriptome from the length of our small intestinal tract, and I'm just showing pictures for full fed versus malnourished. So just by changing the diet, we can see huge changes in genes that occur between our groups. And if we look at what's going on, there's mainly differences in energetic balances. So there's an increase in gluconeogenesis and an increase in lipolysis in the intestinal tract and a decrease in lipogenesis. There's a bunch of genes involved with cell proliferation that are all downregulated. And there's a lot of genes that are involved with responding to stress. So cytochrome P450s and oxidation redox reaction, these are all tending to be elevated. So we're trying to get a picture of how these things are working together to give us our phenotype. And we also have a set of metagenome data. So again, we see distinct, I showed you some other representations of this data of looking at differences between full-fed and malnourished animals. You can see there is distinction between the groups. So our full-fed animals make a lot of short-chain fatty acids, which are really important to the energetics of the gut. They also make a lot of the biomarkers of a healthy gut. And our malnourished animals are making methanogens, and we have species that are involved with inflammation and metabolic disorder. So we're trying to piece these together. In addition, just to make it more fun, we've also added in the liver metabolomics from these same animals. So there's multiple metabolites in the liver that are also dysregulated between full-fed and malnourished animals. Glutathione is one of them, which is one of the key metabolites related to stress. And so basically my student here, Raquel, is trying to integrate all these data sets to give us an idea of what's driving all these changes we see in our animals. And so you can see from this representation, we have blocks of genes and metabolites and bacteria that are either up or down regulated in between our full-fed and malnourished animals. So from this data, we're trying to tease out the systematic, basically fallouts from all these different omics to try to relate it to our phenotype we're seeing in our animal. Okay? And we're also doing this for our milk treatments and all our groups to try to tease out how lysozyme is actually doing what it's doing to our animals. So this is sort of the story of the progression of our work to date. We're also working to make models of inflammatory bowel disease in the pigs. So we've done this by using a chemical method, which is DSS, which is done in a lot of mouse models. You feed this chemical and it basically strips the lining of the intestine. And we're also doing gene editing using CRISPR-Cas9 to try to make models of IBD. So IBD is associated with a dysbiosis in the intestinal microbiota. So we think our milk could help try to restore this dysbiosis in context of IBD. And we're also just this year, we've started a human clinical trial in association with the City of Hope, and this is a cancer research hospital in Los Angeles. And the milk is being used in patients who are undergoing a standard myeloablative hematopoietic stem cell transplant as part of their cancer therapy. So one of the main side effects of the therapy of the stem cell transplant is E. coli-based diarrhea. So they're very interested to see if the milk can help these patients not get as sick from the side effects from their treatment. So this is very exciting for us. It's also a little scary. We're about five on our fifth patient right now who have been consuming the milk during their treatment. So finally, I'd like to end by thanking all my collaborators at UC Davis and other places. We have a huge team that have worked or are working with our transgenic animals. I'd like to thank Kathleen and Stephanie have done a lot of the microbiota work. And Raquel is key at trying to integrate all our omics data. I'd also like to thank the Bill and Melinda Gates Foundation, funded our malnourished work with the pigs. And we've also had several grants from the BRAG program at USDA-NIFA that have gone to characterize our goats. And we also have a current one right now where we're looking at off-targeting events when we do gene editing in pig zygotes. So with that, I'm going to leave you with one last slide, and that's Artemis the goat. And our goal with Artemis the goat is one day we hope that she lives up to her namesake, who's the original Greek goddess Artemis, who was considered the mother of all. And when you see a statue of her, she has mammary glands everywhere, and she's feeding all the animals. So we hope one day Artemis's milk can and will be used in people to help improve their health. And with that, I will stop there, and I'm happy to answer any questions.
Great. Well, thank you so much. That was wonderful. Do we have any questions off the bat? Okay, looks like we have a hand up. Let me see what I can do to unmute him. Can I get some help with that? Which one? Misumi? Okay. Okay, Misumi, if you would like to ask your question, feel free to go for it. Okay, Dr. McGill, if you would like to ask your question, please go for it.
Okay, I was just wondering why, if we have this lysozyme get into our stomach and intestine, why don't we digest it, break it down?
Yeah, so we do. So one of the properties of lysozyme is it's very heat and acid tolerant. So we think it can get into at least the upper small intestine before it's completely degraded by the acid.
Thank you.
Mm-hmm.
Hi, Dr. Maga. Thank you very much for that presentation. I wanted to ask that I know in the beginning you had mentioned that when you were working with the GOAT system, you did that for logistical reasons. And when you were looking at the transgenic levels, it was at about 68%. I think you had mentioned somewhere around there. If you had done that in cows, do you think that you would see in terms, or what differences do you think you would have seen in the amount of lysozyme being produced if you had gone with a cow system instead?
Yeah, so cows, of course, make more milk than goats. So our level of expression, so I should note our animals are heterozygote. So because all our animals came from one founder animal, we waited many generations before we start crossing transgenic animals together. So we've just started doing that. Now we have a homozygous line. So when we did this work in mice, when we made homozygous animals, the level of lysozyme increased in the milk. So we would suspect in our goats, we're not quite there yet, but the levels of lysozyme would go up. And with respect to the cows, so when we do transgenesis with microinjection, the transgene integrates randomly in the genome. So we don't have a lot of control over, we don't have any control. So the level of expression could have to do with where it integrated, make sure it's in an active region of chromatin. And the level of expression could also do with the promoter and elements that we gave to the animals. So we would expect if we had done this in cows to see a similar thing.
Okay, great. Thank you very much.
Okay, looks like we have a Rathor, Kirti Rathor, if you want to ask your question, go for it.
Okay, thank you. Very nice talk, enjoyed it. Couple of questions. One is with regards to the fact that in many cultures, goat milk is considered very beneficial to health. And I was wondering if you had checked the lysozyme level in different breeds of goats. And the second question is, it's nice to see that it's finally being used in human trials, with cancer patients and all that. but is there any plan to take it forward for, using it on young children and seeing if it'll benefit them? And I know there's a lot of other requirements getting this thing deregulated and everything. So I'm just wondering where you are at. Thank you.
Yeah, thank you. Those are good questions. So we haven't looked at lysozyme levels in other breeds. That would be very interesting. The breeds we have here are mostly Swiss breeds, so Swiss dairy breeds, and also a meat goat. But yeah, that would be a great thing to do. And in terms of using it in other places besides cancer treatment, we have worked in the past in northern Brazil. We had a grant through the Ministry of a joint grant with a couple of universities there through the Ministry of Science and Technology to bring the genetics of the lysozyme line to goats in that region. There are goats there, and there's a lot of big incidents of diarrheal illnesses and deaths in that region of Brazil. So we got the goats there through a lot of roundabout processes. So there are goats on the ground, but the grant has ended, and so that's sort of in suspension. But the government there was very excited to try to get this there to help the people, as was the chancellor at one of the universities. So, but as usual, I mean, there are still a lot of qualms sort of against transgenics, and people, don't know if they're safe or things like that. So that's sort of a battle we always have of getting these technologies incorporated and accepted.
Thank you.
I did have a question for you. Whenever you were talking about metabolomics, I was wondering, were you studying select few metabolites, or was this a full metabolomics profile? And if so, did you identify any differentially expressed proteins that were of interest or surprising to you in that study?
Yeah, so when we did the serum metabolomics, is that what you mean? In the goats, we did a non-targeted exploit. We just looked at whatever was there using GC, MS, and GC, TOF, and then mass spec sequencing. And I have lists of, there's several hundred of them that we identified, and there was one that was different, and it was an unknown. We don't know what it is.
Very interesting. Okay.
Yeah, we haven't gone back to mine that data anymore. But
right. That's a lot of data to mine.
it is a lot. Yeah.
Yeah. Awesome. Well, I don't see any other questions. It looks like Brendan has a question.
You partially answered the question with Dr. Rathor's question, but you said that diarrheal diseases are a major issue for children who are at risk with them. Are you dealing with any particular hurdles when it comes to the use of transgenic goats in delivering these kinds of adapted milk to these different regions, or have you been seeing kind of like an open arms kind of approach from different countries that are dealing with this as a major issue?
Yeah, that's a good question. I think people are very interested in doing it, but I think the regulation is sort of I don't know if in the way is the right word for it, but we have special use authorization from the FDA. So transgenic animals are regulated by the US Center for Veterinary Medicine branch of the FDA. So we have authorization for them to use the milk for the clinical trial. But when we start talking about other countries, the milk has to go through their regulatory approval. And when we were in Brazil, it was hard no one knew where to put it. So like the Center for Veterinary Medicine, they approved the animal, right? It's transgenic, that's why they can regulate it. But then the milk, when it goes somewhere else, then nobody knows if that's another agency that has to approve it or what. So there's still some bottlenecks in the regulation part of the thing. But I do think there is an interest in the milk.
Okay.
Thank you.
Awesome. Well, thank you so much. If there are no other questions, I'll give everybody a couple seconds for any last minute hand raises. Okay, well, I don't think so. So thank you so much, Dr. Maga. That was very interesting.